Hi, Edith. How are you?
Good, how are you?
Good. So I heard today that the review session was a doozy.
That was what, sorry?
That the review session was a doozy today. It was pretty tough.
That was a lot, yeah.
Yeah. So did you come out of it with any questions or?
Um, I think what I'm struggling the most is the TSA stuff, the tubes.
Oh, the TSIs?
Yeah.
Okay. So is it more interpretation that's difficult?
Yeah.
Let me pull up those slides. So let me pull up the slides and then let me show you some things that I think would help. Yes, this is the correct one. First try, I got the right PowerPoint. Okay. So here we go. Alrighty. So when we look at the TSI tubes and we want to interpret like these results, it is easier to look at the ingredients first and then worry about what it looks like. So knowing these ingredients will help a lot and where they're located. So we have our protein source first. Now this is going to be our peptones, and those are in the slant. No peptones are located in the butt portion of the TSI. Then we have our phenol red located all throughout, right? And the phenol red will indicate if the three sugars were fermented. So glucose is only in the butt, lactose and sucrose are in the slant. So glucose only in the butt and lactose sucrose only in the slant. And then we have the sodium thiosulfate. This is the substrate for the hydrogen, the H2S. So this is what happens at the molecular level, right? We have the sodium thiosulfate that will be part of the actual reaction with the bacterial. I want to say, what is this? I think it has like a reductase. The bacteria has an enzyme that reduces sodium thiosulfate. So we need the sodium thiosulfate to be present for that enzyme to work. So with that enzyme present, it might reduce the sodium thiosulfate, but there's no way for us to visually see that. So that means that we have to have that ferrous sulfate in there so that it can detect it and form that black color. So we know our ingredients, right? And now we also know that, if a sugar is fermented, it's going to change the phenol indicator to a yellow color. And if it's not touched at all, there will be no change. Or if a protein source was used, it's going to turn into a deeper color because it's going to produce an alkaline byproduct. So it'll turn that pH indicator a deeper color. And we also know now that we have that ferrous sulfate, which detects that gas and turns it black. So we now know we can have a color change, we can have the black, and then also remember we can have the cracks in the agar, which we'll look at some examples here in a second. But now that we know the ingredients, let's go on And we also know where they're located, right? Let's go on to the types of reactions that can occur in the slant and the butt. So the slant portion, that's aerobic, right? And you can even see some bacteria grow on the slant portion on top. But remember, when we stab the butt, that is our anaerobic portion. So this is us testing, does it like to grow anaerobically or does it like to grow aerobically? What does it like to do? Let's see. So I talked about this also, that glucose is in a smaller quantity than the lactose and sucrose. That's a good thing to know. But let's look at some types of reactions that we can see. I'll go back to that one. So here's some types of reactions that we can see. On the far left is our control, right? There's no change. This is the color it started with. This is the color we ended with. So if we move on to number one, right, we see that same color that we started with, but a deeper pink color up on the slant. So what do you think that means?
So it has some sugar in the butt.
So it does contain sugar in the butt, but there was no change, right? The bacteria didn't touch this little area, but we do have, I'll zoom in a little bit if I can. But we do have a pink reaction up here, right? So with our pH indicator, this on the far left, that's what neutral looks like. So we came to a deeper color right here. So what is the pH indicator telling us that's happening in the slant?
Does it have fermentation?
So it does not have the fermentation of the sugars, but it has the fermentation of the peptones. So, well, it's not necessarily fermentation, it's just a catabolic reaction, but they're being used. The peptones are being used. And that produces our more alkaline environment. And that turns the pH indicator that deeper pink that you see.
Oh, okay.
So now compare, so we said that the butt portion right here, there was no change, right? And the slant, we have the alkaline environment right here. So if I were to ask you what sugars does this ferment, what would you say?
For the first one?
Yes, for number one. Not the control on the far left, but this one that we were, the number one. So what sugars were fermented in that first one?
Glucose and lactose.
Just remember that I kind of set you up for a trick question, but there was a no change and then a deeper alkaline color. If anything was fermented, what color would it have turned?
Oh, yellow?
Yes. So in number one, do you think any of the sugars were used?
Oh, no.
No. Okay. So we can see the bottom didn't do anything. And so what was located in the slant that caused this pink color.
Oh, the peptone?
Yes. So this one, we would interpret it as no change or NC slash NC slash A, right? And there was no sugar fermentation, but there was the peptones catabolized. So that's how we would interpret that first one. Now, knowing what you have here, what number one looks like, tell me how to interpret number two.
So number two will have a glucose in the butt.
Glucose in the butt, yep. And what happened to that glucose?
The glucose was able to have fermentation.
Yes, it was able to be fermented, yes. So we understand the butt, and we figured out what was happening in the butt. Now what happened in the slant?
I had the peptone.
Yep, the peptones were catabolized. So how would we write this out?
Would it be Ka, like the top?
Yes. Yes, exactly correct. So remember that we have, so if I were to ask you a question about number two, and I said, was lactose fermented, what would you tell me?
No lactose.
Lactose was not fermented, correct. What about sucrose? Was that fermented?
No.
Nope. So yes, only glucose, right? Okay. So let's go on to the third tube, right? I'm gonna try to zoom a little bit more. Whoa. There we go. All right. What the heck? Okay. I hope that this is helping-ish. So there we go. So number three, what do you see?
So number three has the black butt.
Mm-hmm. Good. And what does the slant have?
It's a little bit of pink, it seems like.
Yeah,
But
you're right.
Minor.
Yeah, you're right. So for this one, remember that if there's ever that black butt, there is a yellow butt underneath it. Always remember that.
Okay. Mm-hmm.
So we have, so if I asked you what sugars were fermented, what would you say?
Glucose.
Glucose, good. And then what would the black butt tell you?
So it has glucose. Does it mean that has another sugar as well?
Not necessarily, because remember that butt only has the glucose, and you told me just a second ago that the slant was pink.
Oh yeah, pink. So it has the peptone.
Yes, but what does the black butt specifically tell you?
Oh, that did have precipitation.
It has the H2S production, right?
H2S. Okay.
Yes, because if you were to look at something like 4 and tell me, yeah, there's gas, then that's correct, because you don't know what type of gas it is, right? It could be CO2, it could be oxygen, it could be any one of those things. But our TSI tube has that special indicator for H2S that turns this butt black. So there is a black precipitate that you can see, but it is specifically showing you that there was H2S production.
Oh, okay.
So then let's look at number 4.
So for number 3, sorry, would it be like KaH2S?
It would be Ka plus H2S, yes.
Okay.
I know the bottom looks a little funky. It almost looks like there's like air down there, but it'll be a little clearer on the exam than this is currently.
Right, okay.
So yes. All right. So then tell me how you would interpret number 4.
So number 4, did the glucose fermentate on the top?
So remember that glucose is always going to be in the butt. So if the butt is yellow, it fermented glucose. But what are our other 2 sugars?
Oh, lactose and sucrose?
Yes, and those 2 are located where?
On the butt?
In the slant.
In the slant, okay.
Yes. So looking at number 2, remember, like you said, this yellow butt meant that it was only glucose. So if we dissected this and said, let's look at the butt only, right? It's yellow. That means glucose was for sure fermented, right? And then what other obvious thing that do we see?
The air at the bottom?
Yes. So we can't say that this is H2S because it didn't give us that black precipitate. But we can say that there is gas production. So we now know our butt and the gas production side of it. Now let's look at just our slant. What happened with our slant?
The slant itself turned yellow?
Yes.
Yellow only, so but I'm not sure if it's lactose or sucrose.
So that's the thing. When we have a slant turn yellow, we can say it's lactose and or sucrose, because you're right, we don't know.
Okay.
Both are in the slant, and we can't tell which one was fermented. We just know one of them at least was fermented, one or both, right?
Oh, okay.
So then we look at 4A right here, right? Because you interpreted this correctly. What is the difference between 4 and 4A right here?
4A has like a hint of pink or red, yeah.
So for that one, she would never give you guys something in between like this. But you would say that you would start to say that the peptones were starting to be used. So what inevitably happened in this too was the bacteria fermented the glucose. It fermented the lactose and or the sucrose in the slant, but it ran out of energy sources. So it resorted to the peptones. And when it resorted to the peptones, it counteracted the acidic products it made by using these sugars and replaced it with the more alkaline product from catabolizing the peptones. So it gives you that hint of pink, because remember, the peptones are still in that slant, even with the sucrose and lactose there. Okay. So she wouldn't give you something like this, not something so, with so many things happening, but trying to look at that and say, okay, there's a lot of things happening together at once is a good thing to practice.
Right. Okay.
So then let's look at number 5. So knowing now what you saw with these last few tubes, tell me what you would write out in terms of the slant slash butt and what that would mean.
So for number 5, it would have glucose in the butt.
It would.
With the yellow undercoat, or
Yeah, yellow's underneath that black butt.
yeah, and then the slant would be lactose or sucrose.
And or yeah.
And I can't tell if it has like a hint of red as well.
For this one, I would just say don't worry about if there's a hint of red, but your what you said previously, 100% correct.
Okay. Can we go over the first one again? Sorry, number 1.
So the number 1 tube right here.
Yeah.
So to the left of it is our control. That's what the C is for. So our number 1 control shows us what it would look like if there was no bacteria introduced into this TSI tube.
Oh, okay.
So if this is our control and we compare it to the other colors that it might produce, with number 1, we can see that between these 2 butts, there's not a whole lot of a difference, right?
Right.
But if we look at the slants, there is for sure a difference, right?
Right. Uh-huh.
So that's what this is indicating, is that there is a change in the slant, but not the butt. So that would be so that would be a K for the slant slash NC, which means no change or no fermentation. So for this one, there's no acidic byproduct, right? Anywhere in this tube. So if there's no acidic byproduct, what does that tell you about what's happened to the sugars?
The sugars were not able to ferment.
So the bacteria didn't touch them. They were not fermented at all. But what was it? Remember what was in that slant, right? It was the peptones.
Yeah.
So if the peptones were used, it changed the color. It made it that deeper, more alkaline color with the Phenol Red pH indicator. Think of the phenol as just like a pH strip. Like it's good to separate the ingredients kind of like that, because if you think of the phenol as like a pH strip, you're going to remember, okay, I'm comparing colors, right? It's not necessarily like, oh, this is just another thing that makes up that jelly in the tube, right? So that's a way to separate out those ingredients.
Okay.
So yeah, this little page it helps a lot to look at it and say, okay, this is what I would see, this is how I would write it out, and this is how I would interpret it.
Okay. And is she gonna give us the tubes? I mean, are they gonna be pictures, or are they gonna be like the real tubes that we can hold and see?
They're going to be real tubes that you can hold and see. And remember, do not be afraid to ask a question. If you were to look at something and be like, I don't know if this is a crack in the auger, I don't know if this is necessarily, gas production, you can call her over and say, is this gas production? I see a crack here. And she picks out the ones that are pretty obvious. So she will tell you, either maybe think again, or yes, that's correct, or like you're on the right track.
Uh-huh.
So don't be afraid that if you're not sure about something, ask her. But they will more than likely be pretty obvious. Just kind of go with your gut. But if you're questioning it, just ask. It's always a safe bet.
Okay. Mm-hmm.
So yeah, and then also another, I know it's a lot of information that not only do you have to be able to look at this, interpret it, and write it out, but it's also an important aspect to understand this side of it as well, because this is what is happening at the molecular level with the black precipitate. I keep on saying that. But this is what's happening, at the smallest level, right? And it helps you to remember, we had our ingredients, right? The sodium thiosulfate is what is being used, remember? So we see the thiosulfate here being reduced by the bacterial enzyme thiosulfate reductase, and we get the hydrogen sulfide gas and the sulfite production. And remember, this hydrogen sulfide gas can be indicated by the ferrous sulfate. Think of this as another type of like indicator, right? This is another, it's not pH indicator, it's just an H2S indicator. So it's in the shadows, it's, you're looking for color with that one. But with this one, you're seeing a reaction.
Okay.
Because, she might ask you, if you see something like number 3, describe what is happening at a molecular level. And you would have to say, the glucose, remember, because that yellow's under the black butt, the glucose is being fermented by bacteria. We see a kind of a reddish pink, more alkaline slant, which means that the peptones are being catabolized. And then at the molecular level, we have H2S production, which takes the thiosulfate from the agar and uses the enzyme from the bacteria, thiosulfate reductase, to produce these two products, in which hydrogen sulfide gas is indicated by ferrous sulfate.
Mm-hmm.
So that would be a complete explanation as to what is happening in this third tube.
Oh, okay. I mean, is that something that they should be looking for, the entire explanation?
Yes. So you'll always want to be thinking, what is happening?
Okay.
What is this telling me? And if you can look at this and say, this is a book and I need to get as much information from it as I can, that'll be your best way to be able to answer questions. The more in depth you go, you're covering your butt more, so to speak.
Mm-hmm.
So if you go into pretty good detail, then there's no way that she can mark you wrong, right?
Right. Okay.
So that's kind of the mentality of like, if you can look at it, it's trying to speak to you, it's trying to tell you something.
Right.
And then translating that onto the exam will help you a lot. And then you can take that information and then answer other questions with it, right? So because I don't think that there's going to be any multiple choice questions about the TSI slants, but there's going to be a whole section on the lab portion that'll be interpret and explain what's happening.
Right. Okay. Okay.
And if you do want to review again, I'll post this recording, but on YouTube I made another little walkthrough, and I kind of went through these same slides, and I said, okay, this is what's in it. This is what is happening at the molecular level, and I kind of just talked about the different reactions that can happen. If you are just like burnt out, can't read anymore, can't write anymore, and you are having any troubles, just pop it on in the background.
Right. Okay.
Okay. Alrighty. So that helped. That was good.
Yeah. That helped a lot. Thank you.
Yeah, no problem.
Yeah.
Is there anything else that you came across that you might need any more help on?
Oh, yeah, the other lab portion about the it was that lab where we did a pregnancy test and the other test to check for
Yes, the pregnancy and the staff techs, right?
the staff test.
So I'll pull those up real quick. But the is it just like, did you have a question about like the antigen versus the antigen versus the, what am I thinking? Those two. Did you have trouble differentiating between I'm trying to think of the word.
So yeah, so I know for the pregnancy test, like we need to know that the antibodies are like on the strip of the test and that it targets for like HCG. And today she said to make sure that B cells is what is produced the on the strip by the antibodies.
So B cells produce the antibodies.
Produce antibodies, okay.
Because remember, B cells are part of your immune system. And another part of your immune system is the antibodies it produces.
Okay.
So, so you told me that on the strip, on the pregnancy test, on the strip was the antibody or the antigen?
The antibodies.
The antibodies. Okay. So let's see. So if there's an antibody on the strip, the other side of that is the antigen, right?
Right.
So the antigen would be in the urine for a pregnancy test.
Oh, the urine. Okay, yeah. And then, okay, yeah. And then okay, yes. So the urine is what has the HCG, right? And so is the urine what has the HCG and then the strip is what has the antibodies?
Yes.
Right, okay.
And then I'm gonna send out a picture of what she drew out in the in lab for the staff techs test.
Okay.
Because you could read words on the page, or you can look at this picture, which does a great job of explaining what's happening. So let me send it out on Remind really quickly so that you can have it.
Okay.
Okay. Alrighty, so if we look at the picture I just sent out, we can see for the staff text test that S. aureus, the bacteria itself, has this protein A on the outside of it, right?
Mm-hmm.
So the protein A on the outside of S. aureus has an affinity to attach to the rabbit IgG. The IgG is an antibody.
Okay.
So that rabbit IgG attaches to the protein A, and when it attaches to the protein A, it forms a complex and breaks off the little parts of it. And then what happens is the coagulase, also released by S. aureus, it breaks down the fibrinogen in the solution that is part of the rabbit IgG. It breaks down the fibrinogen into just fibrin. Let's see. And then when it forms these complexes between the partial rabbit IgG and the fibrinogen, it clots. So let me see if I can pull up. I know that was a little bit of a weird explanation because this is a weird test. Okay. So here, I'll pull up the staff text thing. So S. aureus. Let's look at what S. aureus has first. So S. aureus has this enzyme called coagulase. Now, coagulase, from the root word coagulate, obviously is going to clot or cause something to coagulate, right?
Right.
So S. aureus has coagulase, but it also has that protein A on the outside of its cells. Okay, so that's S. aureus. That's what S. aureus has. Now, if we look at this test, right, all we used was that little solution. The staff text test solution has these latex beads coated with fibrinogen and the IgG. So the staff text test has the fibrinogen, coated beads, and IgG. So moving on from that, let's see. So we know what we have for each one, right? And if you look at the picture that I sent out, we have the coagulase that's being released from S. aureus, and then we have that protein A that's on the outside of S. aureus. And what's coming in is the latex bead, which is that little blue dot, right? And the rabbit IgG is being represented by the Y sitting on top of the latex bead. Now we also have in this mixture fibrinogen coming in. Now fibrinogen is going to be cut down by the coagulase. And the fibrinogen is just going to turn into fibrin. So now S. aureus is interacting with our solution, and it's coagulating. And now this coagulation is sticking to the bead, that latex bead that's coming in. And we have the detection of the coagulase, and the IgG is attaching to the protein A, causing even more coagulation, and it gives us those visible dark blue dots. And that's how we can tell that it's S. aureus, because Um,
Oh yeah, because of the clumps it had on the test we did, right?
And because S. aureus has coagulase and protein A.
Mm-hmm. Okay.
So remember, the IgG plus the protein A is an agglutination, and then the interaction between fibrinogen and the coagulase will turn that into fibrin, which clots. So there's two different clots going on at once, and it's all with these latex beads, and that causes us to have these big, obvious agglutinations right here. So those are like the two things that are happening at once with that test, because because otherwise we wouldn't see enough agglutination if there was no protein A or if there was no coagulase.
Right. Okay.
So that's what's special about staph, is that it has both of those things. And then one thing that you that might be asked as like a trick question is, if you were to see this result on the left, would that tell you that that could be MRSA, or is that S. aureus?
Oh, I think she asked that on the lab. I think it would be just it's aureus, right? Not MRSA.
Yes, because what was the test you did a couple of times with the antibiotics? That
Oh.
Mueller-Hilton, Kirby-Bauer, right?
Uh-huh.
So unless you do one of those tests, you can't see if a bacteria is necessarily resistant, right?
Mm-hmm.
So with this one, all you can tell is that it agglutinated, which means that it has protein A and it has coagulase, which means that it's some type of S. aureus.
Okay. Protein.
And that's just about it for this test. It's relatively simple.
Okay.
And then did you want me to go into more detail on the pregnancy test? Or do you think you got that one down?
I think I got that one down. Is she gonna like show us like a positive or negative test and go from there?
Yes. So she'll say, okay, she might also give you a test that's invalid. So what would look like an invalid test to you?
Invalid will be if the control strip doesn't show.
So if that control strip doesn't show, then the test is invalid, and it can't tell you one way or the other. So good. But it might also be something like, okay, here's, you can look at it, it has the control strip, and it also has a positive strip, right? So yay, the test is pregnant. So if you were to look at that and you can interpret it, that's half the battle. Then turn around and tell us what's happening on the molecular level with what is in the urine and what is on the strip and how are they interacting.
What is in the urine, what is in the strip, and how they interact?
Yes, that's half of it, the battle with most of these tests. Is what's in here, what's here, and then what reaction does it produce and what does it tell you?
Okay. So the urine will be just to review so I can think about it again.
Yeah.
So the urine will have the HCG, and then the strip has the B cells, and that's how they interact to say if it's positive or negative, right?
The The strip itself has the antibodies produced by B cells. Yes.
Mm-hmm. Antibody. Okay, yeah, produced by.
Yeah.
Okay. Yeah, it's a lot.
Yeah, it is a lot of information. I just recommend for a lot of these, I think I've showed this to the group before, but since I'm right here, first of all, these videos are very good for parasite reviews. You'll need to know parasites like the back of your hand.
Okay.
And then down here, if you're still having trouble with any of the labs, go through these, and the document is going to be like a, define this. It'll ask you a question, things like that. But the PowerPoint down here will just be pictures. Most of it's going to be parasites. Those pictures, if you can look at it and tell me exactly what it is, then you have your parasites down. And then you also need to go in and say, okay, what does it do? What is its mode of transmission? It's a lot of information, but these two things are here to make it a little bit easier to figure out exactly what you need to emphasize on. But I do recommend the document a little bit more because it covers every single test, not just the parasites. And I dedicate a whole good couple hours to the parasites. And once you get burnt out from reading about all the parasites, watch the videos about all the parasites.
Okay.
I promise you, I've went through this before. I know how painful it is.
Yeah.
But I know that every one of you guys that are in this class is capable of doing it.
I just had a question on, let me see. I saw it a couple days ago. It was, where was it? Oh. Oh, under exam 3 material, under lab 15 for the parasites, she wrote down like, go to the following chapters to research the diseases covered in class. Oh, I guess that's just like stuff that we could read from the book.
Yeah, basically.
Oh, okay.
It's going to be a whole lot of just fluffier language for what was mentioned in the actual parasites lecture. But if you learn that way and you just need to read it in a bunch of different ways, then that's a great way to look into it more, and just to know a little bit more about it. But if you're more of a visual learner like me, then looking at the YouTube videos, I might even try to send a few in the next couple days, or tonight, about some other ones, some other parasites. But
Okay.
let me show you on here. I made a whole playlist specifically for not with that. I made a whole playlist right here. I know there's only 4 videos, but it's about the different parasites. They are the videos down here, but some of these links just don't work anymore. And I'll try to find better videos to replace them. So stay updated on maybe what I link here. But these are all great videos, and she recommends these. And then over here, there is other review ones. So this is some lab tests, which includes the staff text test. Somebody actually performs it. And then this is some other review videos that are good. That includes some virus stuff and things like that. And then I'll upload PAL sessions here, and I'll also link them on Remind.
Okay.
But yeah. So stay posted on those, and I'll try to give you guys as much information or as many resources as possible to have this be delivered in a variety of ways. So
Yeah, for sure.
it's like throwing like 30 darts at a dartboard and just hoping at least one sticks, because that's all you need.
Yeah. Yeah, it's just yeah, so much.
And then there's this old phrase that I stand by, and it's basically like, don't keep sawing down a tree with the dull blade, because you need to sharpen your blade, right? You need to rest. Because if you keep going at it with a dull blade, it's gonna cost more energy than what it would be worth in the time that you would lose by just taking a break, relaxing for a minute.
No, for sure. Yeah. Right after we finished like class, the review, I took my dogs out for a short walk just to have a little break before I came to your session.
Yeah, there you go. And that's perfect.
Yeah.
So keep finding ways to do that, and you'll be fine. You'll do just fine.
Thank you. Yeah. I really want to end up with like a B in the class. I don't want it to go down to a C. So I'm like on that borderline. So
well, we'll keep going at it, and then if you get lucky, and maybe if you like go on your hands and knees and get Dr. B a coffee from Starbucks, you might be able to get some extra credit points in there.
yeah, yeah, it's true.
But we'll see how the exam goes. And we were even talking, because of kind of how the semester is right now, because this is our first semester, our first spring semester in person again.
Right.
So you guys are the guinea pigs, and we'll be lenient if need be. Like if you guys are just not getting it, if you guys are having trouble with the deadlines, we'll try to work with you. But that's only if you guys are legitimately struggling.
Right.
So I think we're gonna evaluate after this exam. So just take up any extra credit opportunities you can. Make sure you try your best on all the projects, because trust me, that can bring your grade up a lot. With all those, with the micro project, with the senior picnic project. What other one is there? It's micro and your unknown project. All three of those projects are worth so many points, and they can bump you up a lot. I mean, all of those combined is worth more than the final.
Right. Okay.
So
yeah,
try really hard on those. Like after you're done with this exam, like switch gears and knock those things out. And if you ever have questions about your micro project, you remember it has to be kind of specific, but I can always answer any questions you might have, like, hey, is this section need to be more specific? How does this sound? Things like that. I can answer those things if you can make it to any Zoom sessions, or even just text me. I won't guarantee immediate response, but I'll respond as soon as I can. But yeah, just crank those things out after the exam. And I believe you're Monday, Wednesday, right? You're Monday,
right.
Wednesday.
Yes.
So that means that you'll be done with the exam by Wednesday, leaving a solid, 4 days of, you don't have to go to school unless you have other classes, but a solid 4 days of no new micro material.
Right.
So taking that time to focus in will also help.
Yeah, for sure. Thank you. Yeah, for the micro project, I'm doing it with two other girls from lab.
Mm-hmm.
So we were able to do the 3D model during spring break.
That's great. That's really
Yeah.
good.
Yeah. So now we're just working on like, typing everything up for the board. So
Mhm.
maybe I'll ask you about my section if I need to be more specific or something.
Okay. Yeah, and then if you look at the under assignments, if you look for the micro project, you can see the rubric, and if you follow that rubric pretty much to a T, you're almost guaranteed 100%. Like, and then if you guys all want to jump on and do a mock presentation, I don't mind doing that.
Oh yeah, we should. Yeah.
Just to get like the jitters out of just you've said it once before when you get up in front of the class, so
Right. Right. For sure. Yeah. Yeah. It's a good idea. I'll tell the girls. I
okay.
think we wanted to practice with ourselves anyway through Zoom, but if you're there, I think that's better so you can give feedback.
Mhm.
Okay. Yeah.
Yeah, no problem.
Thank you. Yeah, this really helped for those two lab sections that I was kind of stuck on, but
Mhm. Well, if you have any more questions, feel free to text me. I'll also, well, unfortunately, I won't have another review session before your guys' exam. But if you do have any questions between now and then, you can shoot me a quick text. If it's something that a lot of people are stuck on, I might just make it like a quick YouTube video about it and explain it and upload that. But yeah. So maybe go take your dogs for another walk and then
yeah.
or have some dinner and then uh, do a little bit more studying and then get some good rest for another day of studying tomorrow. But
Yeah. Yeah, for sure. Yeah, thank you.
mm, no
Yeah,
problem.
I probably won't go to work tomorrow. So yeah. Okay. Thank you.
No problem. Have a good one.
You too. Thank you.